I'm your substitute teacher this morning. I'm just going to go on where I left off yesterday, and then we'll see how far we get, and then I can go into the next talk to finish off. All right, so the topic I want to talk to you about today is, where does gene regulation come from? So there are a fairly large number of studies about gene regulatory networks and how gene regulation operates. And basically in all these studies, it's implicitly assumed that the transcription factors and their targets have in some way co-evolved, such as to fine-tune how much the transcription factors up or down regulate their targets in each condition. Right? So it's just assumed that somehow the system is doing the right thing. And the question I want to focus on here is, how do you get to such a situation, starting from a situation without the regulation? How does evolution go from something that is totally unregulated to having some finely tuned machine? Because if you think about it, there's quite a number of things that would have to happen. So first of all, some regulatory protein that the cell has, some DNA binding transcription factor, would have to evolve to become sensitive to some signal, right? So you go from a situation where your gene expression state does not depend on this signal, and you want to go to a gene expression state where, to a situation where your gene expression state becomes dependent on the signal, some food or some stress. So first of all, you have to evolve a regulator to evolve dependence on this signal, to sense this signal. And then second, the appropriate target genes, namely the genes that you want to upregulate when the signal is present, they have to evolve binding sites for this regulator, right, so as to become activated by it. And you have to, of course, avoid that any other genes that you don't want to upregulate evolve binding sites. And finally, to make it a well-tuned machine, you would also have to tune how strongly the regulator responds to the signal and how strongly it upregulates each of these targets. And so it would seem that that's a lot of things to happen. So how would this happen in evolution? And so we think we gained some insights into a general mechanism by which this might happen. And these insights came actually from a study where we were studying something else. We were interested in gene expression noise in E. coli. And yes. Sorry? That's what I'm going to first introduce in the next, 8 slides or something like that. All right, so we were focusing on gene expression noise, and then we found that it had some relationships to the evolution of gene regulation. So that's what I'm going to tell you about today. All right. Gene expression noise, what do I mean? So here's a little cartoon of how, in the very simplest situation in bacteria, gene expression works. So some gene here is sitting in the genome. There are these proteins, RNA polymerases, that transcribe. They have a binding site near the start of this gene. And so in conditions where the concentration of this polymerase is roughly constant, there is roughly a constant rate at which a polymerase will come and start transcribing a gene and make a new mRNA. All right? But when there is a constant rate, of course, it doesn't mean it happens every 5 seconds exactly, right? But it's a stochastic, how long you wait between consecutive events where the polymerase finds the start of this gene and transcribes. Then, for each mRNA that now exists in the cell, there is also a roughly constant rate at which a ribosome will come and start translating this mRNA. So there is some translation rate, lambda p, which is the probability per unit time per mRNA that another translation event starts. mRNAs can also decay, so there is some rate per unit time, probability per unit time per mRNA that it will decay. And once you've made a protein through translation, also these proteins have a certain rate per unit time at which they decay. Right, so in the simplest model, there are sort of constant probabilities per unit time per molecule for these events to happen. All right, so let's first look at just the distribution of mRNA levels that you expect to get. Okay, we don't look at the protein yet. So as I said, there is a constant rate per unit time, lambda r, at which you make new proteins, mRNAs, sorry. And each mRNA has a rate, mu r, of decaying. So now, if I want to solve for the probability that at time t there will be n mRNAs in the cell, I can write down this kind of differential equation, often called the master equation. So the change per unit time in this probability is, well, it's the probability that you had n minus 1 mRNAs, and with this probability per unit time, you made another one. This is transcription. Or you had n plus 1 mRNAs, and with this probability, one of them decayed, and now you have n. And then you lose the probability to stay in state n by the probability that you currently have n, and one of them decays, which happens with this probability, or you make another one, which happens with this probability. All right? Now, if you look at the steady state of this equation, it's very simple. It's a Poisson distribution. So in steady state, the probability to have n mRNAs is given by this Poisson distribution, the transcription rate over the decay rate to the power n, 1 over n factorial, e to the minus, again, this ratio of rates. The mean of this distribution is just the ratio between the transcription rate and the decay rate. All right? The variance of this distribution is also the transcription rate divided by the decay. So that's one of the key features of a Poisson distribution, that the mean and variance are the same. And so the standard deviation is equal to the square root of the mean of the number. So here are some examples. This is when there are on average 0.1 mRNAs per cell. So there's probability of 0, 1, 2, 3. This is when it's 1 on average, and this is when it's 10 on average. Okay? When the number becomes big, this looks roughly like a Gaussian, but as I will show you in a minute, in E. coli, you're much more often in this regime than you're in that regime. All right, now we want to add translation and protein decay. So we want to look at the level of the proteins. Okay, so remember again, we have this transcription rate, decay rate per mRNA, the translation rate per mRNA, and the protein decay rate. Right. To solve this distribution in full generality, I don't think there is actually a closed form, but there are very, very good approximations. So for example, proteins are often much longer lived, right? So these rates are typically much bigger than this protein decay rate. And so you can make an assumption that the protein decay rate is slow relative to the other ones, which is a very good assumption in almost all scenarios. And then you can analytically solve. So for example, this was done in this paper. And then it turns out the limit distribution that you now get in the number of proteins per cell is a negative binomial distribution. And it depends on two parameters, A and B. So the parameter A is the number of transcription events. So it's the transcription rate divided by the protein decay rate. So you basically can think about it like how many new transcripts are made during the lifetime of one protein on average. As we will see in a minute, in many situations when bacteria are growing exponentially, the protein decay rate is so much slower than the growth rate of the bacteria that the effective rate at which the protein concentration goes down is in fact just the growth rate of the cell. It's dilution that dominates the loss of proteins, not decay. So in that situation, this rate here is effectively the dilution rate or growth rate of the cell. And so this parameter then means how many transcription events do you have per cell cycle on average? All right, so that's what this A means. And the B, which is sometimes called burst size, is the translation rate divided by the, that's an error, this should be mu r, sorry, by the mRNA decay rate. And that's the average number of time an mRNA is translated before it disappears. Right, so you could, they call this a burst sometimes because you think they make a new mRNA, then you make a burst of proteins from this mRNA, and then the mRNA decays. And so this is how many proteins on average you get per mRNA. Now, if you look at the mean and variance, the mean number of proteins is just the product of these two parameters. And the variance is the mean, but now multiplied by this burst size plus 1. All right, so the difference between the Poisson distribution, where the mean was the same as the variance, now the variance is b plus 1 times the mean. So effectively what is happening is that this translation is amplifying the fluctuations in mRNA number. And the amplification constant is b. All right, so the bigger the burst size for mRNA, the more proteins you get, and the more variable the protein number will be. I'll also make one side remark. So for example, is that if you want to keep the mean of your protein equal and lower its noise, what you should do is you should downregulate this B and upregulate this A. All right, so you want to make an mRNA that is badly translated, decays very fast, but you transcribe it all the time. All right? And so once these kind of results became clear, there were some people that knew about regulation of microRNAs. And so as you know, in higher eukaryotes, microRNAs are important for regulation, but they're kind of mysterious because when you knock them out, you don't see very big changes when you look at bulk expression of population of cells. And people have been very confused about that. And so at some point, people start proposing maybe what microRNAs regulate is not the mean levels of proteins, but the noise. And microRNAs exactly target this number B, because they'll lower the rate of translation and they increase the rate of degradation of mRNAs. Okay, that's what they do. And so this is something, I'm mentioning this as a side remark, so there are some recent works that have shown that that is probably really the case, at least in some situations. And I'll just mention it because it very naturally flows once you understand how sort of this burst size controls the noise at the protein level. Yeah. Mm-hmm. Oh, you mean how do these rates, so the protein, I'm not quite sure. You mean how is the mRNA? Okay, so the transcript, I'm looking now at the situation where these rates are assumed to be constant. Okay, so they're not affected by anything in this model. No, now this is proteins. We went to the protein level. N is integer. Yes. 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 Yes, exactly. It's mean times this B, essentially, right? B plus 1. And so the noise, if you look at the standard deviation divided by the mean, which is also called the coefficient of variation, so that's the square root of the variance over the mean squared, you will see that that's B plus 1 over N. Square root of B plus 1 over N. Okay. So highly expressed genes will on average have lower noise than low expressed genes. And there will be this factor B, that says genes that make very big bursts of protein from each mRNA. They have more noise than genes that make small bursts. All right, okay. So now I just want to give you some sense of what the numbers are for all these things in E. coli, this in the bacteria, just to have some sense of what numbers, what regimes of the parameters are we in. So the way you can do this is you can make probes that hybridize to mRNAs, right? So they're antisense to the mRNA sequence that you want to measure, that have fluorescent lights on them, fluorescent molecules on them, I'm sorry, And then when you add such probes to the cell, you can basically take pictures of cells and count the number of spots that you see per cell. And this number of spots is the number of mRNA within a cell. This is actually easier said than done, as you have to work quite hard to get these things right and so on. But in this way, you can really count the integer number of mRNAs per cell and get a distribution for each gene of your interest of how many mRNAs per cell are there. And yeah. What will not depend on mRNA size? I have not said anything about it. All I have said is they're constant. So for a gene, there are these 4 numbers, and I assume that in sort of steady state growth, these numbers are roughly constant. And I look at what will happen if they're roughly constant. And how those numbers depend on all kinds of things, like Sanjay was also asking, how does it depend on other protein concentrations? So I have not talked about this yet. I just want to say, let's just look at the consequences of assuming they're constant. All right, so what are the copy numbers of mRNAs per cell? So here's a histogram of how many genes there are. This is from a paper of Sunny Xia's lab, where the log 2 mRNA copies per cell is shown here. All right, so you see that the most common mean number of mRNAs per cell is -2. So that on log 2, that means a quarter. All right, so the most common case for a gene in E. coli is that there is 1/4 mRNAs per cell. That is to say, 3/4 of the cells have no mRNAs, and 1 in 4 has 1. That's actually the most common case. You see that it's actually rare to have more than 1. And there are genes that have, an mRNA once every 60 cells. Okay, so the sort of typical situation for not a high expressed gene in E. coli is you sit around without any mRNA, wait. Okay, now you get 1 mRNA, then you get a burst of proteins from this mRNA, and then your mRNA is gone again, and you sit with your proteins, and then as the cell grows, sort of their concentration slowly dilutes out, and maybe 1-2 generations later, you get another burst of protein. Okay, so this is a very noisy regime, right, that you sit in. Okay, so some additional numbers. These RNA polymerases, right, that transcribe all the genes. So E. coli has about 4,500 genes. There are, depending on growth rate, something between 1,500 and 10,000 of those per cell. And the ribosomes that translate the mRNAs into protein, depending on growth rate, there are somewhere between 14,000 and 45,000. These mRNAs, here is some data from a paper of Bernstein et al. Of 2002. Their decay rates are between 1 and 15 minutes. So the most common is something like between 3 and 5 minutes, and it ranges up to 15. Okay, so they turn over fairly fast, mRNAs. They don't live very long. And the protein number, so you see that here in pink, this is the distribution of mean protein number. This has a peak at 10. And it goes down even to guys that have less, 1/10 of a protein per cell. And the most highly expressed one have 1,000 to 10,000 proteins per cell. Okay, so these guys are some of the highest expressed proteins in the cell. Most proteins have much lower numbers, and many proteins are somewhere between 1 and 100. Okay, so there too, you're in a regime where you expect all kinds of finite size fluctuations to matter most of the time. Okay? So there's all kinds of reason to think that gene expressions would be very noisy. And it is. Okay? So it's a miracle that E. coli manages to survive. If I sort of step back and think about it, I would think this is a mess. No? All right. So how do we actually measure gene expression noise? So one of the sort of first seminal papers in this area is a 2002 paper by Michael Elowitz and Peter Swain. Well, what they did is, so this circle here is the chromosome of E. coli. This is the origin of replication. And they went a roughly equal amount of distance to both sides, and they stuck a red fluorescent protein here in the genome and a green fluorescent protein there in the genome. And these two genes have the exact identical promoter. So they're controlled by the same promoter, so all the sort of transcription regulation and so on should be the same for these promoters. And then you just put this construct in the cell and you take pictures. And so you get pictures like this. So what you see here is that when these two proteins express at roughly equal levels, the cells look yellowish, all right? Because there's both red and green in same amounts and they look yellow. And so the total intensity of light from cell to cell, how this fluctuates from cell to cell, tells you how much gene expression fluctuations there are from cell to cell, but you can also look how the color changes from cell to cell. So you can look at the relative levels of green versus red, and that tells you the different amount of expression of these two identical promoters in the same cell. All right, and basically what they showed is that the total variance, namely the variance in the amount of green per cell plus the variance in the amount of red per cell, you can basically decompose in the sum of two terms, which they call the intrinsic variance and the extrinsic variance. And the intrinsic variance is basically the average squared deviation between the amount of green and red. All right, it's basically the average difference between the levels of these two. They call that the intrinsic variance, because it's like how much different is the expression of two identical proteins with identical promoters in the same cell. So they say this is like an intrinsic noise that you apparently can't explain by any of the states of the cell. And then the rest of the variance they call the extrinsic variance, and that basically tells you how much variation there is in the total intensity across cells. All right, so another way you can think about this is if you think this is the amount of green per cell and this is the amount of red per cell, and each dot here is a picture of an E. coli cell where they measure the amount of green and red, then the intrinsic noise, or the extrinsic noise is the noise along the diagonal, where the variance along the diagonal where the green and red are the same, and the extrinsic noise is the component of the variance that is orthogonal to this diagonal. All right? Okay, so what they found is that as you take a promoter, and I don't want to go into details, but they had some way that this promoter was regulated by a transcription factor, and so they could change the level of this transcription factor by adding something to the cells. And so they could induce this promoter. So they could make it go from a low expression level to a high expression level. So that's what they did here. So here the average fluorescence of the promoter goes up. And here is the noise, right? So this is the standard deviation divided by mean. And you see that this intrinsic noise falls roughly like 1/n, as we expected when you have this constant rate of transcription. All right, so that's what they see. This goes down. But the extrinsic component of the noise, which is shown here in green, this jumps up and then comes back down as you upregulate the gene. All right, so as you go from a gene that is transcribed at a low rate and you upregulate it by now activating some transcription factor, this extrinsic variance, this variance from cell to cell goes up. So why is this? Where is this extrinsic noise coming from? Well, one thing that is important to realize is that we've basically been assuming that this promoter is transcribed at a constant rate, but that's not true in reality. And in particular, when promoters are regulated by transcription factor, the promoters can be in different state, right? So assume that this promoter here is regulated by one activator protein, one transcription factor that activates it. Then this promoter can exist in three states. One with nobody binding, one with the RNA polymerase binding, it's actually 4 states. One with only the activator binding, and one with both the activator and the polymerase binding. Right? And the idea is that when the activator is binding, the rate of transcription is higher than when the activator is not binding. So basically what's happening is that the promoter is stochastically switching between a state where the activator is not bound and a state where the activator is bound. And when you're in a state where the activator is bound, you're transcribing at a higher rate than when the activator is not bound. This will increase the noise. And if the level of this activator, the concentration of this activator fluctuates from cell to cell, then the amount of time that the promoter is going to be in the bound state also fluctuates from cell to cell, and this will lead to the increase of extrinsic noise. All right, so that's our yes. Yeah, but I didn't say that they have to sum, did I? I mean, the total variance will sum, but this is noise. That's variance divided by mean squared and then taking the square root. So that thing will not sum. All right. Okay, so this... all right, so this is how this started. And so since then, a lot of people have been doing these kind of measurements of using these kind of fluorescent reporters to measure gene expression noise. And one lab, the lab of Uri Alon, so this was done by Saslaver et al., they made a large library where they took every promoter in E. coli and they put it upstream of a plasmid with GFP. All right, so there is now this library that you can use to measure the gene expression noise of any E. coli gene you want. Right, so what they did is, but when you have the intergenic region between two genes, they cut it out and they put it in front of GFP. Okay? So what you can then do is you can put these plasmids in the cells, and assuming that the GFP fluorescence per cell is proportional to protein number, and we know that this is a good approximation, you can then measure the distribution of the amount of GFP per cells in cells. And you can do this in high throughput using a machine called FOX. And so this is actually what we did. We started characterizing the gene expression distribution of every E. coli promoter. So a FOX is a machine where cells are made to flow through a flow channel one by one. So here you see the cells coming down this flow channel. Then there is a laser that activates the fluorescent proteins as they pass in front of it, and a detector that measures the amount of fluorescence that is coming from the cell. So you can now measure the amount of fluorescence per cell, and you can do this in high throughput. In this FOX, you can have like 1,000 cells per second or something, I forget, but it's really fast, yes. Just tell me stop. This is mean protein number. Yeah, this is from single cell. Well, okay, this was done at, these were integrated in the chromosome and not on the plasmid. So it's using very similar techniques. Like you can also just make pictures of the cells and measure the fluorescence from microscope images. There are different ways of doing it, but so I'm now telling about this way that we use to do it for all promoters in E. coli. Right, this was done not for all promoters in E. coli, but a relatively large number. It was a very big study. All right. So, and I actually think the date is wrong. This is not 2022. I think it might be 2012. I will check it for you. Okay. So, it's a FOX, and one thing that you should know is that FOX actually stands for fluorescence activated cell sorting, is that the FOX has the other nice property that depending on the amount of fluorescence it measures, it can charge these cells with an electric charge, and then they move in front of this electric field, and so they will move in different directions depending on their charge, and you can use that to sort your cells into buckets corresponding to different expression levels. And so this will be important later on. All right, so here's an example of what we see when we do this. Okay, so these are two different promoters. We also made pictures in the microscope of what a colony of cells looks like with this promoter. And so you see that this promoter is less noisy than that promoter. And I picked these two examples because they have clearly different noise, but they have roughly the same mean. Okay? And if you now go to the fox and you measure like 50,000 cells and you plot a histogram of the log of GFP expression, okay, so this is a histogram of log expression, they look Gaussian to a very, very good approximation, right? And this is actually the case for the vast majority of promoters. It's actually hard to find promoters where this does not look Gaussian. Even like a Kolmogorov-Smirnov test with 50,000 points, half of the time you pass it. Okay, so for biology, this is close you get to a Gaussian, I would say. So you can characterize the distribution of each promoter by two numbers, the mean and the variance or standard deviation. So that's what we did. And so if you do that, you get a picture that looks like this. So along the x-axis is the mean of the log expression of the cell, so that's this mu. And along the y-axis is the variance minus a lower bound on the variance as a function of mean. So the slide was taken out, but basically what you should, as I showed you before, at any expression level, there is always, at minimum, this Poisson fluctuation noise. And this scales like 1 over the mean. Okay? And it turns out that in the damn Fox machine, there is a lot of shot noise from the measurement, and that adds another term that goes like 1 over the mean. Okay? Those two things that depend on the measurement have been removed. All right? So this intrinsic part that you, lower bound on noise that you never can go below, has been removed. So this is the excess noise, it's the amount of noise, the amount of variance beyond that. All right? So you see many promoters here seem to be bunched up near the minimum that is possible. But there are some promoters that have much higher noise. So now what we were interested in is how has natural selection in evolution acted on these noise levels? Right? Because this noise level is encoded in the sequence of the promoter. When we do these measurements, the only difference between the cells is the small piece of DNA that is sitting upstream of GFP on the plasmid. Other than that, everything is the same. So these differences in noise must come from that sequence. Okay? So somehow that means if it's encoded in the sequence, natural selection can act on it. And so we wanted to explore how has natural selection determined these noise levels. So for example, you might imagine that noise in gene expression is like inaccuracy. And so you might want to minimize this. And maybe the fact that many of these guys are bunched up here is because selection has sort of optimized the promoter to minimize the amount of noise that it has. And then maybe these are just genes where you don't care about the fact that they're noisy somehow. Or maybe there is some reason that for some promoters you actually want to be noisy. Okay, so we wanted to answer this question. And to answer this question, it would be very useful to know, okay, what kind of noise would an E. coli promoter have if you didn't select for noise? Right? You would like to have some reference. What would it look like if you hadn't put selection on noise? And yes. Yeah. Mm-hmm. Two promoters that are identical, they will be at the same point. Because that's the same construct. But I would not even be able to tell you that these are 2 points. I mean, that's just doing the experiment twice. Right? Because when you give me a promoter, I put it in front of GFP, and then I stick that into the cell, and then I measure. So if you now give me again the same promoter, tell me to measure it, I'm going to do the same thing. So that's just, you're asking me about the replicate measurements. Mm-hmm. No, each of these dots here is the mean and variance of a distribution that may involve 50,000 cells. Right? So so the way this works is you make these plasmids, you put them in the cells, Yes. So we have a stack of 96 well plates, and in each of these little wells on the plate is a solution in which E. coli are that are carrying one of these promoters. And there are 96 on one plate, and we have about 20 of these plates. Yes. Yes. About 2,000. Actually, there are not 2,000 points in this picture because there are promoters that are so low that we can't detect them. And they're not here. So it ends here at 7 because below there is mostly background fluorescence that you're measuring, so we cut it off. Yes. 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 But that was exactly this experiment. No? That's exactly what they decided to do, yes. Yes. I'm really confused. This will be just the total variance. It will be just the total variance of one of the two promoters. This is the variance across cells of a single promoter. No, they will have the same variance. But within one cell, they won't have the same expression level. Right. So that's the point, right? So here they made a green and a red, right? These are identical promoters. And so if you ask about the variance of the red across many cells, or the variance of the green across many cells, that will be the same, and that's what I'm measuring. But if you look at one picture, you will see that some cells are greenish, and some cells are reddish, and some cells are yellowish. So the relative amount within one cell are different. Yes. Exactly. But I'm doing it for one, I'm not doing it for a pair. I don't. Okay. All right. So to know about how selection has acted on these noise levels, what we decided to do, yes. Doesn't matter. It's the favorite question to be asked in this talk. I've never passed once giving this talk without somebody asking me this question. So this is a low copy number plasmid that has on average 6 copies per cell. You can do this on the chromosome. I mean, the absolute levels are all lower, but qualitatively it's all the same. And it's in fact the case that, when you put something on so people always worry, well, isn't the plasmid copy number also contributing to the noise because it fluctuates? But on a chromosome, the chromosome copy number also fluctuates across cells and across the cell cycle. And in fact, I have now reason to believe, after studying this for a while, that the plasmid copy number may actually be more constant than chromosome copy number, relatively speaking. Okay? So that concern, I think, is a red herring. That the plasmid may look more noisy than the chromosome. Doesn't look like it. Okay, so what we did is we took the same plasmid, exactly like before, But instead of putting known E. coli promoters upstream, we made a library where we put about 150 base pairs of random DNA. So we had a company make random oligomers of length 150, and we made plasmids containing random. So we had an initial library that had about 1 million different random DNA sequences upstream. We put them all in cells. Right? So we put all these plasmids in cells. So now we have a population of cells where each cell is carrying a different promoter, and the promoter is completely random DNA sequence. Then we go to the FOX, and we set some target level. So we picked some target level, and one time we picked a level that corresponds sort of to the mean expression level of E. coli genes, and once we picked one that corresponds to a very high expression level. But so we pick a target. Then we look at the distribution of fluorescence of the single cells of this population. So there's many cells, all carrying, well, some carrying the same promoter, but many carrying different promoters. And then we measured, then we selected in the FOX the 5% of cells that was closest to the target level. All right? So we set a gate in the FOX that 5% of the cells would make it through, the ones closest to the target level. Right. Then we take the plasmids back out of the cell. We in fact cut these little pieces of random DNA of the selected cells back out of the plasmid. So there are little sites in the DNA where we can have enzymes that cut these things out. So we now have a pot in which we just have these inserts that were selected. And then we do error-prone PCR amplification. So we amplify them, but with an error rate where we expect on average 1 mutation per 100 base pairs. Then we put those onto fresh plasmids, and we put them into fresh cells, and then we go 5 times around the loop like this. So we evolve promoters for some particular expression level like this. And it's important to realize that the reason we go through all this trouble of taking these things back out and putting them back in fresh cells and so on is that we want to make sure only this sequence can evolve, right? Because otherwise, E. coli will find all kinds of ways of mutating other things in the cell that will change the expression level, and it's not in your promoter, and then you're in trouble, right? So in this way, you can make sure it's only in the promoter. All right, so what does this look like? All right, so I'm now showing you the initial library, which we call round 1. What is the distribution of its expression levels? And so this actually is mostly autofluorescence. The distribution that you see here, the peak here is just autofluorescence, but there is a little bit of expression here in the tail. So we were asking for this expression level, a relatively medium level for E. coli genes, so sort of the median of all the E. coli promoters, native E. coli promoters. After then, the selection said any cell that is in this window will make it through. That's 5% of the population. And after doing mutagenesis, this was the distribution of expression levels we got. So you see, we now already have a hump of genes that are expressing near the right level. So this is after 1 round. But we still also have guys that look like they're not expressing. In the second round, this was 5% of the population. Then we got this, and we're pretty much already there. Okay? So 2 rounds of selection and mutation got you there. And then we went 2 more rounds, but not a lot was changing. Okay? So this was the distribution of the fluorescence after in the population. Then we said, okay, we take the 98th percentile of all E. coli genes. This is some ribosomal protein. It's very high expressed. And so this is the evolution that you get then. So it takes longer to get there, but it's not hard. In a couple of rounds, by round 5, you have most of the cells in your population expressing at this very high level. And we have here guys in the tail, we've gotten clones out where we have E. coli promoters that express higher than any known E. coli promoter. Okay, so it's easy to make from random DNA. We started by completely random DNA. Right? Sorry. Same from the same library in both cases. Yes, so this round 1 is exactly the same in both. All right. So now realize that what we have here is still a population of cells that have many different promoters in them. So we wanted to look at individual promoters, what are their mean and noise. So the student that was doing it, she isolated 400 clones of single cells. So what you can do is on a plate, you can streak out things at such low density that the colonies that grow up all grew from a single cell. And then if you take from the colony and grow them up in the tube, now you know all the cells here have the exact same promoter. So we made 400 such clones, and we put those in the FOX and also in the microscope. And here I'm showing you some histograms of the expression distributions of those promoters. So here are examples of some mean expressing promoters, and here are some examples of high expressing promoters. All right, so now we can measure the mean and variance of all these synthetic promoters that we selected, of these few hundred. So now we asked, how do the noise levels of these synthetic promoters compare with those of the native ones in E. coli, that E. coli has evolved over billions of years? And so to our surprise, so these are the red dots here. So the red dots show you the mean and excess noise variance of these synthetic promoters. And we had really expected that these promoters were going to be much noisier than these E. coli promoters that have all this time to be selected. But in fact, if anything, we saw the opposite. All right, so all these promoters are near this lower bound. And if you look at the cumulative distribution of noise levels, so you see that at the lower end, they're very similar, but then clearly the distribution of noise levels of these synthetic promoters is lower relative to the native promoters in black. So somehow, native promoters are enriched for high noise promoters that you don't see for synthetic promoters. Now, okay, so when we saw that, the first thing that we worried about is maybe our selection scheme that selects this 5% every round is implicitly selecting for low noise. So we've not only selected for the mean level, but we've also selected for low noise. It actually sounds quite plausible. Yes? No, because we're asking for one mean, right? That would be an issue if we were asking for different means and comparing different means which have different intrinsic variances. But if you're asking for one mean, then this excess noise is on top of the minimal amount that you can't avoid, right? So, okay, so I don't want to go, because it would take too long, into all the things we did to show that this is not the case. But I can tell you that this is not the case. So you can do additional rounds of selecting 5% over and over. The noise levels don't go down. You can ask about what is the relative, we can really measure what is the fitness function in our facts and measure what is the probability for a promoter to be selected as a function of its mean and its variance, and see if it's plausible that we, in these few generations, could have selected for the guys with the low means, and you can't. Okay? Essentially what is happening is you're selecting out from the initial library those promoters. There's random sequences that give you expression at the right level. Okay? Because we find that actually that we could have done without the mutation. This is one of these things, like the student wanted to kill me afterwards because it was an enormous amount of work to do this, taking the things out and amplifying them and putting them back. But it turns out actually in our final round, most of these sequences, you have whole clouds of sequences that came from a common ancestor in the first round, but there are sequences from the first round that express at these levels. So basically what's happening is you're selecting from your pool of 1 million or so things, those ones that express at the right level, and you're not really having a chance to further separate them by their noise level. And so these low noise levels is what you get by default if you just ask for random sequences that happen to express at this mean level. Okay? There was a question, yes. Okay, so I think we are not measuring gene expression using sequencing. We're measuring gene expression using the fluorescence coming from each cell. So the PCR is something that's totally separate from the gene expression measurement. All right, so now that means if low noise is what you get by default if you ask for a constantly expressed protein, a gene, then it means that those native promoters that are up here must have somehow experienced some selection pressure that pushed them up. Okay? Maybe they weren't directly selected for noise, but something else, and they got higher noise as a side effect of the thing they were selected from, but there must have been some selection pressure that caused these promoters to increase their noise. Okay? I think this is actually one thing that is kind of useful to think about. It is, if you want to know something about how selection has acted in the wild on a trait, noise in this case, it's very hard to tell anything about that. But so here in this case, what we managed to do is we managed to make a situation where we looked at how does this trait behave when you don't select for it. And this allows you to tell at least something about how selection must have sort of acted on this in the wild. All right, so selection must have acted to increase the noise levels of some native promoters. So what is special about native promoters that show high noise? All right. So there again, I want to make a side remark. In biology, so maybe you're sort of the default way of thinking that you've learned about science is that when you look at variables that have nothing to do with each other, they should be uncorrelated. And when you find a good correlation, it means these things really have something to do with each other, right? If you take variables in biology, properties of cells, I would say it's almost the opposite. It's very, very hard to find pairs of variables that really don't correlate at all, right? So you find weak correlations of this noise level with many, many things. Like if you ask, how often does this protein occur across E. coli strains? You'll find some weak correlation. If you ask, how high is it expressed? You'll find some weak correlation. You find correlation with lots of things. But I believe that the main correlation that is dominating in this case is this one. Promoters that are noisy have many regulatory inputs. So there are databases that I talked about in my first talk that tell you about known transcription regulation. And so what you can do is you can sort your promoters from the low noise ones to the high noise ones, and you can calculate the mean and standard deviation of the number of regulatory inputs of promoters, of the subset of promoters that are over a certain noise level. So here on the left, I'm taking all promoters, and this is the average number of regulatory inputs that they have, so it's somewhere below 1. And then as you start taking subsets that have higher and higher noise, systematically the number of regulatory inputs goes up. All right? Here's another way of looking at it. You can look at what is the cumulative distribution of noise levels. So in green, that's for our synthetic promoters. And now, instead of showing it for all native promoters, I divided the native promoters into those that have no known regulatory inputs. So there is no known regulatory inputs for the black ones. For the blue ones, there's exactly 1 regulatory input. And for the red ones, they have 2 or more regulatory inputs. And you see that the genes in E. coli that are not known to be regulated, they have actually noise levels that are fairly close to our synthetic promoters. Whereas those that have regulation systematically have higher noise levels. Okay? So what it says is, if you're a promoter that is constitutively expressed, not regulated, you're going to be low noise. And when you have higher noise, it's likely coming from some gene regulation. So there is a general association between noise and gene regulation, and we would like to understand why this is. And second, somehow we still want to understand what happened, what did selection select on to cause these noise levels to increase. Yes. Step-like kink. Where? In which plot? Oh, this means nothing, right? I mean, these bars are standard errors. This. Ah. Oh. I don't know. I've never really given much credence to this. So, okay, I would have to think about that. But I strongly suspect there's nothing really very interesting going on here. I think that what's happening is that our measurement of noise level is itself noisy. All right, so some of the variability in this regime is not real, right? And so that might, so there is really like, I had to work very hard to make it this clean. Right? This is why it's also it's a strange plot, right? Because what I'm doing is I'm looking at the mean and standard error for all promoters, and as you go to the right, I'm looking at a smaller and smaller subset where I'm cutting it off at some minimal noise level, right? And so what you're seeing here is that for some reason the average number of inputs of the very lowest ones is still below the ones that are low, and then for a while the average number of inputs is not changing, and then it's going up for the ones that are above something. I think it has something to do with this noise in the measurement of the noise, but I had not thought about it before, but it's not important for the talk. Let's put it like this. I have no idea. All right. Is the number of different transcription factors that have binding sites in that promoter. Absolute number of, yes. It's the mean across all the promoters that have this noise level or more. Okay? All right, so now I want to do some theory to understand what's going on. So now it's going to get real fun, okay? Because the interesting stuff, I think, is going to come now. So I first wanted to know what is sort of the fitness function in our Fox machine. So imagine that, so a promoter is characterized by its mean expression level and a standard deviation sigma. And as I said, it is in log scale, so I'll do everything in log scale. It's a roughly Gaussian distribution. So imagine this is the expression distribution of my promoter. Okay. What will be now the fitness of this promoter? Well, okay, what's the fitness function? The fitness function is the Fox says only cells that lie in a certain range make it through selection. And according to the manufacturer of the Fox, this selection is like a little block, right? It's like if you're over this threshold, you're selected, and if you're over this one, you're no longer, right? You have to, but it's not really like that, right? So we've done measurements, calibration measurements that show us that it's actually very well approximated by a Gaussian-shaped peak, where the probability to be selected is highest at the center of this window, and it has some width tau. Okay, so that's roughly also a Gaussian function. The probability that a cell with expression level X will be selected depends on the center of my selection window, mu star, and this width tau, and it's this Gaussian form. All right, so what will be now the fitness of this promoter? It will be simply a convolution of these two functions, right? So I have to do this integral. And because these are both Gaussian shapes, those integrals can be done easily. And so you can think of it as the fraction of this population, of the blue distribution, that falls inside this yellowish window. Okay, and that fraction is given by this expression here. All right, so it's given between the difference between the mean of the promoter and the target level squared, divided by the sum of the variances, namely the width of the target level and the width of the distribution of the promoter. All right, so what does that look like? It looks like this. So what are the properties of this fitness function? So remember again, this is the fitness of a promoter that has mean mu and standard deviation sigma when I'm asking for target level mu star with a tolerance of tau. Okay? So the first thing is that if you have a promoter that has the wrong mean, you can always increase your fitness by putting your mean closer to the target level. All right? So when mu of the promoter moves closer to mu star, as in here, this area under this curve will always go up. Okay, so that's sort of obvious, but I'm just pointing it out. Now, once you're at the optimum, right? So once you've optimized your mean, so you've centered your promoter to be at the target level, you can further increase your fitness by lowering your noise. And you will basically have a selection pressure to lower your noise until the entire population of your cells falls inside the selection window, as I've seen here. All right, so when you have optimal mean, it's good to lower your noise. Again, it's intuitive. But if you're at the wrong mean, you might not want to lower your variance. Right? So here I've looked at a promoter that had the wrong mean, and it had this overlap with the selection. But now I've lowered the noise of this promoter, and actually the overlap has gone down. Okay, so it turns out if the mean of your promoter is more than a standard deviation away from the optimal level, then actually it starts becoming a bad idea to become too low noise, because it will lead that nobody in your distribution will intersect your window. All right, so so if you want to say this sort of fancily, there is a bifurcation. As the expression, the deviation between mu and mu star gets bigger, the optimal noise goes from 0 and at some point starts increasing. And beyond this level where it was 0, the optimal noise level is actually the square deviation between your mean, the target mean squared, minus this width coming from the tolerance, square root. Okay. So now I want to look at a situation where I'm going to imagine that what happens in the wild is that this E. coli cells, they live in one environment, and the fitness of a promoter is really a similar function. It's basically saying your reproduction rate and survival rate is maximal when you express at some optimal level, and this reproduction rate lowers as your expression level goes away from this optimum. But as E. coli is experiencing different environments, this target level is changing in time. So for one time you have this target level, then you have that target level. So what you should think of is now you have a situation where, for some period of time the selection asked for this, then it asked for this, then it asked for that. And now you can first ask, what will be the fitness of a promoter that is not regulated? I start with not a regulated promoter with mean mu and standard deviation sigma. What will be the fitness of this promoter when it now experiences all these different environments? And you can show that the log of the fitness across all these environments is simply the average of the log of the fitness in each environment. And if you work it out, then you'll see that it's given as the mean of the promoter minus the mean in an environment squared, and this averaged over all environments. Right, so this is the average distance squared between the mean of the promoter and the means across the different environments, divided by the sum of this tau squared and sigma squared. And then here is another term that depends on the tolerance and the variance. All right, so if I have an unregulated promoter, if I don't allow myself regulation, what would be the optimal mean and variance for this promoter? Well, the optimal mean would be the mean of your promoter is actually the average of the levels that the environment is asking you for. So mu is the average of these environmental levels, target levels. And the variance, the optimal variance, is the variance in these target levels across environments minus this width squared. That is to say, you place your mean expression in the middle of what the environments are asking you for, and you make your variance wide enough that you ensure that some members of the population intersect every environment. Okay? That's the optimal solution. Now, in the sort of evolutionary literature, this is often sort of referred to as a bed hatching scenario, right? So you know you're going to get different environments that ask you to do different thing, and you make a very noisy distribution such that there are some members in your population that satisfy each of these requirements. Now the question is, okay, but wouldn't it be better to evolve gene regulation rather than to... Okay, so how would that work if you evolve gene regulation? So again, I'm looking at this environment. Here's the unregulated genes distribution. These are the 3 target levels, but it works for any kind of distribution of target levels, right? I'm just doing this for illustration. So this is without regulation. Now, ideally, there would be a transcription factor in the genome whose expression is upregulated in the green condition and downregulated in the red condition, right? So imagine there is a transcription factor whose expression in this gold condition is like this, in the green it's like that, and in the red it's like that. If I now evolve a binding site in my promoter for this regulator, then what will happen is that my mean will be shifted up by the regulator in this condition, and it will be shifted down by the regulator in that condition. And these will be now the target levels. These will be the expression levels of my target gene in the 3 conditions. And you see this is an even better solution because there is a much higher fraction of cells intersecting what you want. Okay, so this is just saying gene expression regulation will work, right? There's a point for it. But I want to now stop for a second and make you realize is that when I do this, right, when I couple the gene, the target gene, to this transcription factor, 2 things happen. First of all, the mean of the promoter's expression level becomes a function of the mean of the transcription factor in that condition. I call that the condition response, and that's how we typically think of gene regulation. When the transcription factor goes up, the target goes up. When it goes down, the target goes down. But in every condition, the transcription factor itself will not be infinitely accurately expressed. It will also fluctuate in its level from cell to cell. Okay, so there will be some noise in this regulator, and that noise will necessarily propagate to the target genes. Okay, so these distributions here of the target gene are per definition wider than they were when they were not regulated, because there will be propagation of noise from this transcription factor to the target. All right, so this is noise propagation. And so what I did is I actually worked out a general theory how the combination of these effects now affects the fitness of this promoter in these conditions that I talked about here. All right? So, I mean, I won't do the derivation here, but I can tell you it's really not so hard. All you have to assume is that the fitness function in each environment is roughly Gaussian shape as a function of expression level. And then you allow it to vary in any kind of way, and you can calculate the fitness across all these environments. And it turns out, so in this theory, that the fitness of coupling a target promoter to a regulator depends only on 4 effective parameters. Okay, so there are 4 effective parameters. The first one is which is I call the expression mismatch, and it's the variance in the desired levels across environment. So how much does the target level vary across environments? Divided by the width of the expression distribution of the unregulated promoter. And so it's basically saying how many standard deviations away from the target level is the promoter on average. So it's a measure of how unhappy it is when it's not regulated. The second one is the signal-to-noise level of the regulator. It basically measures how much does the mean of the regulator vary across environments relative to the width of the distribution of the regulator in each environment. So if this is high, the regulator is very accurate. If this is low, the regulator is very noisy. The third one, and this is a very obvious one, is how well do the levels of the regulator across conditions correlate with the target levels across conditions? So is the regulator going up and down in the way that you desire, right? So there is just a Pearson correlation between those two things. And finally, how strong do you couple? Right? So as the regulator increases twofold, is your target going to go twofold or fourfold or eightfold or only 50% higher? So there's a coupling strength. So as a function of those four parameters, you can work out what is the logarithm of the fitness in terms of these four parameters. Yes? It's all in these averages. So when I say variance, then it's a variance that you calculate with the distribution of how often each of these environments occurs. All right, so it's this expression, it's some polynomial in these four parameters, and there is some term that is here logarithmic in the square of the coupling. So I'll just, okay, so it's not so intuitive what this now predicts, so I will show you with a simple example. I will start with a promoter that's unregulated and is fairly unhappy. So this number here, y squared, is I think 16. So it's on average 4 standard deviations away from where it wants to be. And now I'm going to ask, if I couple this promoter to a regulator that has a certain signal to noise and a certain correlation with the desired levels, and I optimize the coupling strength, so I tune the coupling strength to be optimal, what fitness can I get to? Okay? All right, so that's this. So along here is the signal to noise of the regulator. Here's noisy regulators, here's very precise regulators. Along here is the correlation between the regulator's levels and your desired levels. So up here, the regulator is perfectly matching what you want. Up here, the regulator is going, down here, the regulator is going up and down in a way that doesn't correlate at all with what you want. Okay, and so now I'll look at 3 example scenarios. So and the color here, so it's a contour plot, is the fitness. So highest fitness is here, lowest fitness is there, and these lines are lines of equal fitness. So here's one scenario. So the best situation you can have is if you find a regulator that has very high correlation, like 0.95 with what you want, with the desired levels across environments, and is relatively high accuracy, so high signal to noise. So here is a cartoon example of such a situation. So the promoter is, the transcription factor is going up and down in the way you want, and so the target level of the promoter goes up and down in the way you want. And the noise level of the promoter is only increased a bit due to noise propagation. So in this scenario, all the benefits that the regulator gives you are from this condition response effect. And the noise propagation that comes along with it is really just a deleterious side effect that you can't avoid. Okay? However, if you go over here, I have a regulator that has no correlation whatsoever with the desired levels across environment, but it's a very noisy regulator. So what it will do is it will just essentially broaden the distribution of the target levels and implement this bed hedging scenario. But that's already a lot better than no regulation. Okay, so in this scenario, you also get an increase in fitness coupling the regulator to the target promoter. But here it's coming entirely from noise propagation and not from the condition response. And what is even more surprising maybe is that this situation here can outcompete a situation where the transcription factor actually has a reasonably good correlation with what you desire. So here, the correlation is 0.64 across environments, but the fitness that you reach here is actually lower than there. So why is that? How can it be that a transcription factor with higher correlation with what you want still give you lower fitness when you couple to it? The reason is that this transcription factor is simply not noisy enough. Its signal to noise is so accurate that it's putting you exactly at some position, but that position is still the wrong position, and you will have too low of an overlap, at least in 1 or 2 environments. So in fact, if I were here and I would actually make this transcription factor more noisy, I would move over here and increase my fitness. So now you can ask, okay, so let's now ask, as a function of my correlation, what would be the optimal noise level of a transcription factor to have? Okay? How noisy would I want my regulator to be so as to optimize my fitness? And that's this red curve here. All right, so you see, when you find a regulator that's very, very accurately doing what you want, has very high correlation, then you want it to be also very high signal to noise. But when the best correlation you can find, if any transcript, let's say, so if you're this target gene, you're looking for some regulator in the genome to surface you. And if there is no regulator whose levels across environment have a correlation of 0.9 with what you want, if the best thing that you can find is 0.7, then you like this regulator to actually be quite noisy. All right? But in this scenario, you're actually already pretty close to the optimal fitness that you could get with a very, very accurate regulator. All right? So, the summary of this theory is that noise propagation is often functional. You know, it helps you increase fitness, and you can almost think of it as a rudimentary form of regulation. If you don't know exactly how you want to be regulated, it already helps to be noisy. And now, what you can imagine is that maybe initially, you couldn't find any regulator that had high correlation with what you want, but you could find a regulator that would make you more noisy. So, in order to evolve gene regulation to de novo, you can maybe start by coupling to a regulator that simply increases your noise. And now, once you're here on this curve, you can imagine a scenario where in evolution, the regulator undergoes mutations to change its expression levels across environments to increase this correlation with your desires, and at the same time, become a little less noisy, and you can, in this very smooth way, move along this curve where noise propagation and this conditioned response are really sort of acting in concert, and end up in this situation up here where you have now very accurate regulation. Right? So this shows you how there is like a smooth evolutionary path from going from no regulation to very accurate regulation, using regulation through noise propagation as an intermediate. Okay? Yes. Yeah. I'm not quite sure I understand the question. You mean this looks like it intersects this minimum? Yeah, I mean, it is true, right? So at any point, this signal to noise lies at the optimum of fitness along this direction. Yeah. Well, it can't reach it. Okay, so there is going to be some, okay, so maybe this is not clear. This optimal signal to noise level has a pole, and it will diverge at some finite correlation. And here that's 0.9 something, and there the thing will diverge, and it's no longer possible, or let's say the noise level that is optimal is 0. Right? You want an infinitely accurate regulator from that point onwards. Right? It's that bifurcation that I showed you earlier. It's the same one that's happening here. Yeah, so you can do things and say these are real phase transitions and so on, but I don't think this is now. The one thing that I want to say, because I see I'm out of time, is I wanted to relate this to something that I talked about the other day. Remember that I showed you that, okay, this is 2 slides and done, that people notice that different proteins evolve at different rates. Different genes in the genome, right? Some genes will go very slow and some genes go very fast. And they showed that the best correlate we have been able to find so far of this is gene expression level. Genes that have a high mean expression level, they evolve slowly. Genes that have a low mean expression level, they evolve fast. So I wanted to know, how do other properties, namely like how noisy is a gene, how plastic is it, how much does its mean level change across conditions? How many regulatory inputs does it have? How does that correlate with these other variables? So what we did is, for E. coli, we collected a whole set of statistics for each gene, namely the rate at which amino acids are substituted, the rate at which synonymous substitutions occur, promoter conservation. So this is, we looked at the intergenic regions, right, the promoter regions, and looked how conserved they are in evolution. So this is telling you something about how fast is regulation evolving. The average protein level, the average mRNA level from our data as well, the number of regulatory inputs, how much does the mean expression of the promoter vary across conditions? So we took a large database of microarray experiments in E. coli to look for each gene, how much does its level vary across different experiments people have done, and finally the expression noise. And so I took that table, so I now have a whole bunch of statistics for each gene, and I did PCA. Okay, so principal component analysis. This is just 2 examples of, you find these sort of weak correlations between lots of things. Okay, so this is mean, this is this expression level against substitution rate. And here, for example, is a correlation between expression plasticity, how much does the gene change expression across conditions, and promoter conservation. How fast is the promoter turning over in evolution? So now when you do principal component analysis, it turns out, okay, so this is the fraction of variance explained by the different components, that the first component has on one side this evolutionary rate of the protein, and on the other side, the RNA level and the protein level. And so that's the thing that we saw before, right? That genes that are expressed highly evolve slowly, genes that are expressed lowly evolve fast. And so it turns out that the second principal component that you find exactly captures this regulatory thing. So on one end of the scale are very high promoters with many regulatory inputs, and typically also a lot of variation of their expression across conditions, where on the other end of the scale is promoter conservation. That is, how conserved is the promoter. So on one end of the scale are promoters that are very noisy, they have many regulatory inputs, they change their expression across conditions, and their promoters evolve fast. Their regulation turns over in evolution. At the other end of the scale are genes that have constant expression across conditions and have low noise, and their promoters also don't evolve very fast. Okay? So I think that this sort of analysis, it captures really an orthogonal dimension of this variability that you find across genes. All right, so with that, I've come to the end for this lecture. Yes? Okay, so promoter noise is the variation in expression levels of single cells in one environment. Plasticity is the variation in the mean of the entire population of cells across environments. I just take E. coli. This is a measurement. And I grow it in some medium. And I measure the expression for a gene, and not just in single cells, but the mean of all cells. Now I change my environment. I add some stress. I add a bit of antibiotic. Okay, now they respond, they do various things. I now measure again for that gene, what's the mean expression level. Now I give them another kind of food. I again measure. And so I do many, many different environments. And in each environment, I calculate the mean expression level of this gene. And that variance, this variance of the mean of the population across environments, that's expression plasticity. All right. Yeah.
A system where there is a unregulated gene expression.
Unregulated or?
Unregulated and regulated gene expression. But I want to know if, in some genes can be expressed to generate different isoforms of the enzyme.
So all I did here was in E. coli, and so generally in bacteria. And so the amount of different isoforms of proteins is, let's say, limited relative to eukaryotes. Okay. So we did not look at that, but we also really totally focused on transcription regulation. So what we did is we took the promoter of a gene and put it in front of a fluorescent reporter. So the thing that we actually measured, the fluorescent reporter, this is always the same one in every construct. So the only thing that is really differing is the promoter. So we're really looking at transcription regulation here.
But suppose I have a system that expresses a particular gene that gives rise to isoforms of the enzyme. In trying to look at this situation, is it good to sum the rate of translation of that particular messenger RNA as one entity, or do I consider them separately?
So good for what? I mean, you say, is it good to consider them one or multiple entities? It depends what you want to do, no? Good for what? What do you want to do?
What I am looking at is, suppose I want to consider this system.
Yes.
And I have a gene expression that led to a particular messenger RNA. Mm-hmm. So if I want to look at the entire system, maybe to arrive at how the final equation on how to predict its, I mean, what you have just described. So I'm thinking, so I'm asking, this rate at which the particular that same messenger RNA is being translated to isoform A, isoform B, isoform C. And is it good to consider them separately in that system, or to sum the rates as one?
Okay, so when we go to sort of, let's say, eukaryotes and that have a lot of different isoforms, then of course there is additional regulation going on at the level of with what fraction of the transcripts that you make will make what splicing isoforms. And so you can also have regulation at that level, and you can, for example, also have noise propagation at that level and variation. So I focus today on transcription regulation, but it's certainly true that there is all these other levels of regulation, right? So you can regulate the mRNA splicing, the mRNA decay rate, and you also have regulation of the protein decay rate, right? So one of the things I might mention is there's very little study of control of protein decay rates. And as far as I can tell, the main reason for that is that we don't know how to accurately measure it. But if you play theoretical games, like if I want to fast and accurately regulate the level of my protein, actually that's the level at which you can most beneficially regulate, right? Most efficiently regulate on a short time scale and accurate, you would attack the protein decay. Like, so you just make enough of it and then you lower it to the exact amount you want using decay, right? So it might very well be that actually protein decay turns out to be the most important level to regulate the precise levels. But we don't know this because we don't know how to look at it. Right? It's like we look where the light is, not where necessarily the keys are. Yes.
So how variable are the copy numbers of kind of global resources, such as RNA polymerase or ribosome? You mentioned about the copy number variation of ribosome, right?
Yeah.
So how variable are those numbers?
I think it's still about 10% or so.
Okay, I'm raising this question because the gene expression level depends not only on these copy numbers, transcription factors, and other small molecules, but also depends on the variation of those 4 rates, the rates from central dogma. It also depends on some hidden variables we cannot keep track of, right? So probably one of the hidden variables, in my opinion, might be those global resource informations.
Yeah, so I totally agree with you. So in right, so we know. So I skipped this sort of part of what is the minimal noise level of any promoter as a function of its mean. But there is, even for the highest expressed genes that are unregulated, some noise level beyond the Poisson noise. So there is some CV, right, that they all have, so even very, very high. So it's a variance that scales as the square of the mean. Okay? And I believe, like you, I think that noise is coming from fluctuations in basic things like RNA polymerase number, ribosome copy number, maybe nucleases that decay mRNAs, and also just fluctuations in the total growth rate, which basically control fluctuations in the dilution rate of your protein. But basically all genes are suffering from those fluctuations, right? They all feel them sort of in the same way. And I focused here on the differences between genes that only depend on their promoter. But I totally agree with you that for the global picture, those fluctuations are very important.
Okay, just
follow up, yes.
Follow up. So it's purely my speculation. Okay, is it possible for numerous genes to suffer from kind of competition for the global resources? Like we have, let's say, 100 copies of RNA polymerase. So more than 100 genes are waiting for RNA polymerase to come. Then this situation may give rise to extra noise.
Very good question. So you're saying, are we in the regime where the ribosome and polymerase copy number is the limiting factor, and basically all ribosomes and all polymerases are busy transcribing and translating all the time, and there is competition between genes and mRNAs for getting them to come to them, right? Or are we in the regime, it could also be that there's many ribosomes and polymerases standing around with nothing to do and waiting for a gene to go to. Those are two very different regimes. So I don't know for a fact what regime we're in. If you're asking me to speculate, I bet we're in the first regime. I'm in the regime where basically every polymerase and ribosome is most of the time busy. And it's really the relative attractiveness of different promoters and different mRNAs that controls how much each is going to be transcribed and translated. That's what I believe, yeah.